Hello, everyone. I'm here with Niklas Freund, or Niklas Friend for our English-speaking audience. Um, Niklas is a geneticist and biochemist from currently working in Cambridge. He's originally from Germany, but before I spend too many words on him, let's have him give us a short introduction. So, Niklas, who are you and what do you do all day?
Who am I? Hi, Manu. Thanks for having me. I'm really happy to be here. Yeah, as you already so eloquently put, I'm Niklas, and just for the audience, the two of us, know each other from our time in Heidelberg together, and yeah, since then I've ventured into new lands, and I'm currently doing a PhD in Cambridge, UK, and just finished my first year, and I'm working at the Medical Research Council Laboratory of Molecular Biology in a lab that I will probably tell you more about during the course of this conversation.
I hope so, indeed. So your PhD is in biochemistry, roughly speaking. So what does it specifically mean? Like, how does your research day usually look like, and what are the topics you're currently looking at?
Okay, so yes, it is biochemistry in the broadest sense. Actually, it is a subfield that's quite hard to define. Um, so basically, I am a chemist by training who has then ventured into the bordering areas of biology, to put it like that.
Hmm.
Um, and there's more to it than just the biochemistry. The field would be called synthetic biology. So basically, what we do is we synthesize and build biological systems, or systems that look like our biological systems, and equip them with function that their natural counterpart might not have, or try to rebuild the functionality of their natural counterpart. And the system that I'm working with is DNA, or nucleic acids in the broadest sense. And it is based on the fact that we know DNA and RNA in all living organisms. And we are trying to build unnatural nucleic acids, or xenonucleic acids, and try to make them work a similar way, and or equip them with different biological functions than their natural counterpart.
So you mentioned you're building synthetic living systems. So and the kind of the foundation of what you're looking at is the DNA system, which is at the core of all living systems on Earth, from the smallest bacteria to mammals and human beings. So obviously DNA is a pretty sophisticated, complicated system, but can you still give a short introduction or like a short overview what actually constitutes DNA and what function it has in living systems?
Yeah, DNA is known to most people as our substance, our molecule, our system of genetic information storage. And the closely related cousin, RNA, has slightly different biological functions, but almost looks the same way in terms of its basic chemical constituent parts. So both of them are um, nucleic acids, or to be more specific, ribonucleic acids, which means they are long chain molecules that consist of repeating building blocks. And every building block carries an information bit, chemically encoded, a moiety, a building block that contains one bit of information, and bits quite literally meaning. And by chaining these bits of information, building blocks, together to a chain molecule, you get a sequence of information, which we know as our DNA sequence, our genetic makeup. And that's exactly the same thinking and the same inspiration that we take in our lab we also work with building blocks and chaining them together to chain molecules that look almost like DNA and RNA.
Yeah, so there are several layers of analysis that you can take a look at DNA with. So, for example, you can look at the very basic atomic level or chemical level. What are the what's the chemical makeup? What kind of molecules are you looking at? But then you can also move up the ladder and look at the functional kind of story behind what makes up DNA. And you mentioned DNA is literally a information storage where you can actually have things like bits on the system. So that's DNA in a very abstract sense. But then if you move up, I guess what you guys are doing is to try to understand more what function the molecules have that you're taking a look at, and what higher order functions could arise from that. So can you, from the perspective of what you are doing, explain and in which sense kind of the information is stored in DNA, in what kind of substances and what kind of molecules, and in which sense maybe you have some kind of freedom in choosing which substances this information is stored in?
Yeah, that's actually a good question, immediately alluding to what we change about DNA to make it unnatural to make these xenonucleic acids, or XNA as we call them. So yeah, they are separate layers of complexity, and I should probably move up the ladder from small to large to explain where we're actually coming in and where other people are also working on. So our genetic information molecule, DNA, looks just like any other chemical substance, which is an obvious fact to us, but I think it's very much worth pointing that out, because that immediately means that by taking atoms, as a chemist, speaking as a chemist, taking atoms and bonding them together so that they resemble the building blocks that we have in our bodies, we can essentially synthesize the same molecule that then will have the same function. So at the most fundamental level, there are atoms which make up these organic molecules. And these organic molecules are the building blocks that I alluded to earlier. They are like Lego bricks that you then stack on top of each other to make Lego towers. And every one of these individual Lego bricks is called a nucleotide. And a nucleotide is composed of 3 different parts, essentially. One that is a ribose sugar, basically a platform that forms the core of the chain. And attached to this ribose sugar is what we call a nuclear base. And a nuclear base is yet another organic molecule that codes for the information sequence. And we have 4 different nuclear bases that can be attached to this ribose. There are more, but for the sake of simplicity, I should say that in our DNA sequence, we have 4 different building blocks and 4 different nuclear bases. So they look different by the way the atoms are bonded together, and they have a different physicochemical appearance, which makes them interact with other molecules that read the sequence. And every nuclear base has a different surface. So a reader, basically a reading molecule, would see all of these 4 nuclear bases in a different light, basically. And then the third part of these nucleotides, of these building blocks, is the linkage. So that all of these ribose sugars, basically the core of the chain molecule, can be actually chemically chained together. And that's a level of complexity that I'm already working with. So I am looking at a nucleic acid that has modifications, chemical modifications at this fundamental level, at the ribose sugar, at the fundamental units of the chain molecule. And then by tweaking every single building block, so every Lego brick, I make essentially a foreign chain molecule. So if we look at DNA and envision that DNA is built of red Lego bricks, then in our body, in every cell in our body, we have towers of red Lego bricks. Whereas in my project and in my lab, we have blue and green and purple Lego bricks, and we want to stack these Lego bricks to make towers that are uniformly green or blue or purple, and they then have a different function in many ways. So that would be the next level of complexity. That's what you alluded to earlier when you said the function of them, the information content. So that's then already approaching biology and crossing the boundaries between chemistry and biology. And we are doing that. We're crossing these boundaries then. So whereas a chain of red Lego bricks might look a certain way, and two DNA chain molecules form the infamous double helix that many people know, a chain of green Lego bricks might behave differently and just have an ever so slightly different appearance and ever so slightly different properties, which makes it fold into a slightly distorted double helix, slightly distorted surface and a 3D structure, basically. And then because of that different appearance, you will have different biological functions. So that's the last layer of complexity that we're working with, the most biological layer of complexity, where once you have this foreign-looking molecule that you build like a chemist, then you can do things with it in a living context, in a cell, not quite a human cell, but let's say in a context where it interacts with other molecules, and you can equip it with function that our natural DNA would not be able to do. So that's what I would call the last layer, the biological layer, the information content.
So yeah, you mentioned that DNA is composed of these base pairs that basically serve the same function as letters of an alphabet, and they look chemically slightly different, so that when there's kind of a mechanism trying to read out what they are trying to say, read out what they mean, then they encode a specific kind of information that is unique and means something to the organism. But it's still not obvious to how you come from this kind of information that's stored in the genes or in the DNA sequence, how you get from that sequence to living systems. So you already mentioned that it's kind of as the top layer of complexity, like the intersection between the chemical, biochemical foundation of life in the form of DNA to living systems. But can you elaborate a bit more how the mechanism connects DNA to proteins and then to neurotransmitters and these kind of things?
Oh yeah, absolutely. So that is then moving away from the realms of chemistry and the complexity level of chemistry. So now I will probably, for the sake of simplicity, stop talking about atoms and nucleotides and stick to the analogy of building blocks and the chain molecule, because essentially, if you want to look at it from a more abstract level, every building block is one bit of information, as I said earlier. And so we have four bits of information that DNA can put at every point. So every point in this chain molecule can have an information content stored in one of these four building blocks. They're called A, T, G, and C. So that's the fundamental code that encodes for information in all living systems. A, T, G, and C. And by chaining together A, T, G, and C building blocks, you can get a sequence ATGC, ATAC, GTAC, and so on and so on. And the longer the chain is, the more variations of information, of individual information bits you can have, obviously. So look at this as the fundamental alphabet with which you can make words and sentences and ultimately an entire instruction manual. And that's exactly what's happening in all living organisms. These building blocks, A, T, G, and C, make different words and sentences which would resemble our genes. And all of these genes taken together, all of these sentences taken together, make up our instruction manual. So let's say you want to determine eye color of a human. Then there has to be some fundamental agent, some fundamental molecule in this human that has the instruction manual for their eye color. Let's say this human has green eyes. Then there must be some page in this long instruction manual that says, give this person green eyes. And this sentence, give this person green eyes, is composed of letters. And now I just said it in Latin letters, but I could just as well translate it. And I mean, translate in our language way of speaking, and translate it to another language, translate it to ATAGCTATG, and so on and so on. So this string of information bits will then code for give this human green eyes. So that would be this entire sentence would be what we call a gene. So gene is still a very fundamental word, a word that can be understood in a very molecular way. It's basically just a long string of letters on our DNA that then codes for some phenotype like eyes. So what is happening there in that cascade is the following. Our DNA is coding, or our DNA has this sequence of information bits stored on it. Then this sequence has to be read by certain molecular machines and transcribed into another chain molecule, which is called RNA. And DNA and RNA look very similar. They're just slight modifications, chemically speaking, but they, on a biological level, they have the same mechanism of functioning, I would say. Not the same. I mean, biologists would probably disagree now, but looking at them as a chemist, they store information in the same fundamental way. So DNA is transcribed to RNA, and then RNA, or in this case called messenger RNA, does exactly that. It is a messenger. It is a middleman, kind of bringing two parties together, and one party is the genetic information, and the other party is the protein that ultimately has biological function. So I'm already mixing up, while I say that, all of these different biomolecules, and that's exactly the fundamental way how the central dogma, as we call it, works. So the fundamental way in which information is read and translated in humans and in basically all organisms that we know. So the middleman, RNA, the messenger, is then translated to a protein sequence, to a string, another string again, a string of amino acids that make up a long chain molecule, which we call protein. And on this cascade from DNA, transcribed to RNA, translated to protein, all of the information is read. And this protein that's then produced might then have an involvement in the green eye color. So kind of a long introduction into molecular biology that I just did, but essentially we go from DNA to RNA to protein, and there are different machines that have different jobs in this cascade.
Exactly, and proteins can serve a lot of different functions in the body. So you mentioned eye color as a very visually appealing example of just seeing that there's a certain chemical substance that has a blue color or a dark color, and then that is read out in the part of the genetic code that encodes the cell types that are used in the eye of the human being that's being born. And then there's a certain molecule that codes for kind of like certain structures in the iris, and then that tends to lead to this emergent property of blue eyes, for example.
Exactly. So it isn't that simple. It isn't that we produce one protein that suddenly has a blue color or a green color. That's not how it works. But for the sake of simplicity, products of this cascade will eventually lead to the phenotype of eye color. But there's more involved, as you just already said.
But again, it can be very interesting to see that literally one letter mutations. And maybe you can also mention a bit what, in which sense mutations can change what's going on there. But it's interesting to note that mutations can actually, like, even minute changes of one letter in this huge genetic instruction manual, that this one letter mutation can completely change how a certain enzyme works, or a certain neurotransmitter, for example, works, or certain receptors of neurotransmitter work. And there's, for instance, there's a lot of diseases that are actually caused by these single mutations that can have pretty horrific influence on the lives of an entire human being. So it's in a sense, it's not always really obvious, like how you can't really say this is a gene for blue eyes, but you can see this is a gene for a certain neurotransmitter, or like certain structure that really, even by this tiny change, can have a huge influence on the biological system at large.
Exactly. If we don't want to talk about it in such an tangible level like eye color, just use that as a very easy to understand genetic trait that is inherited. So everyone realizes how eye color is inherited. But there's much more that's inherited, like, for example, the functionality of a certain neurotransmitter or the functionality of a certain enzyme. The fact that certain people cannot digest alcohol properly is due to the fact that their alcohol dehydrogenase, this enzyme that breaks down alcohol, is not functioning the same way as in other individuals. So on that level, it's also very easy to understand the inheritance of traits that vastly change the molecular makeup of a certain person. And you already alluded to diseases that are due to single point mutations, and there's a whole host of these diseases, and they people like to talk about them because it's easier to understand them, whereas the vast majority of diseases is due to a very complicated, intricate network of genes and mutations that all work together to produce very complex traits. Certain diseases like sickle cell anemia, for example, are really due to one point mutation. So sickle cell anemia is a disease where red blood cells are distorted in their shape to make a sickle shape. And there is a certain mutation on a genetic level, on a DNA level, that's understood and well researched, well understood, that is leading to this phenotype. And the reason is because single letters have such a huge power in that instruction manual. It's a very symbolic language, I would say. Whereas in the Latin language, you have many letters, and changing them in the Latin alphabet, sorry. In the Latin alphabet, we have many letters, and changing one letter might change, let's say, certain grammatical phenotypes of a certain word, but it might not change the entire meaning of the words. Whereas on a genetic level, it can actually do that. So, as I said, it's very symbolic. So every letter has every letter has a certain meaning in that alphabet, and three letters taken together, they code for one amino acid in the final protein product. And that link is purely symbolic. Actually, you can think about it analogously, or you can think about it in an analogous fashion to Turing machines, where every information bit, every letter has a certain linked function, a certain linked phenotype, if I want to stick to the biological word. And in the same way, our genetic code is very symbolic, and it even has the same commands like start and stop. So basically commands that tell the machine head that's reading this code, or in our physiological case, the molecular machineries that are reading the code, tell them where to start, tell them where to stop. And because these triplets of letters have a symbolic link, or are immediately linked to the product, changing one of these letters can link them to another product, which changes the protein product, which then changes the biological cascades in which this protein product is involved in, and so on and so on. So the downstream effects are vast when you just change one letter in this instruction manual.
There were some very interesting and important points in there about genetics and medicine of the future, but I think before we move in that direction, I want to trace back a bit, a couple of points that you mentioned earlier. So first of all, you mentioned you are working in synthetic biology, which is different from the traditional approach to biology in that you are trying to build these living systems synthetically in the lab. And the idea behind taking this approach is also for you to test out in which sense DNA is chemically or just structurally really by necessity the way it is, or in which sense there's still a lot of freedom and leeway in the way you could construct this machinery. And you also mentioned that the letters of the DNA are actually pretty arbitrary, and they are purely symbolic in what they code for, in the same sense that in German or in English, words mean something specific, but you could imagine using different words, which is the whole reason why you have different languages, because you can say kind of the same thing in a lot of different ways. So, and you your lab or kind of your research is moving in this direction of trying to understand in which sense DNA is really has this kind of flexibility, or you could have living systems and genetic machinery that works differently. So what can you tell us about this?
Exactly. So that's moving down the ladder again, back to the complexity level chemistry, on this ladder of the nucleic acids, on this ladder of the systems that we're talking about right now. So I should say to understand the field, the traditional disciplines of biology and also biochemistry, they try to understand a biological or a biochemical phenomenon by deduction and by analysis. So you break down the physiological process that you're trying to analyze, or you break down, sometimes quite literally speaking, the biomolecule that you're studying, and you analyze by whichever means the constituent parts, or you analyze changes to the process that might explain to you how this process works. Whereas the young field of synthetic biology is trying to do exactly the opposite. So we are not breaking down DNA or nucleic acids to understand their function. We're not trying to manipulate genetic circuits to understand how the reading of this alphabet works. But what we are actually doing is the opposite. So not by deduction and analysis, but by synthesis and reconstruction, synthetic biology gains insights. And because our system, our love interest of research is nucleic acids, we try to build nucleic acids in order to understand more about nucleic acids. So people always say you cannot understand what you cannot build, for example, and that's exactly the thinking behind synthetic biology. You try to understand something about the fundamental nature of our genetic storage system by building a genetic storage system, or you try to understand something about why DNA exists and nothing else. Like no other chemical system that stores information. And you try to answer that question by designing and building these alternative, unnatural storage systems and try to see in the process of doing that what makes our DNA so special. So it is actually a very weird fact to think about, but all organisms that we know, and of course, concomitantly, all of them are on this Earth, and would be interesting to see if it's different on other planets, provided that there is life. But all organisms that we know code their genetic information in ribonucleic acids, and the structure of these ribonucleic acids, the structure of DNA, was solved by Francis Crick and James Watson, actually here at this very place in Cambridge. And so after we have discovered the chemical makeup, the fundamental structure of our genetic storage systems, of our universal storage system, we then immediately had to ask the question, so why? Well, so what? We've just found this, so why is that? And so that's exactly what we're trying to do at this very place. And I like to think for that reason, of course, there are a couple of decades in between, but I like to think that exactly for that reason, we now have to step back and think about why we only know ribonucleic acids and why they look the way they do. So yeah, what we're trying to do is design and build artificial nucleic acids, artificial storage systems. And in the process of doing that, we do get an idea of what makes them deficient or what makes DNA superior, or maybe sometimes even realize that these xenonucleic acids actually could serve the same function. They are also capable of being chained together and store a sequence of information that can then be read out by molecular machines. So the question is still around. So what? So why did they not arise? And that's where we come in.
So I guess there are also competing theories about whether DNA just was the one kind of arbitrary gateway that nature took to realize selection, but there could also be kind of what you are also trying to figure out, if DNA just has an evolutionary advantage over all other possible implementations of this machinery, and that DNA just proved to survive in the long run and just dominate all living systems.
Yeah, exactly. That is alluding to another type of evolution. So when the broad public thinks about evolution, or when more biological research labs than ours think about evolution, they think about the journey that nature underwent from the common ancestor cell that was very simple but already living and reproducing to today's organisms and to us humans, for example. And that is biological evolution. That's how sequence and information evolved over time and became more and more elaborate and different to give rise to all of these different organisms. But before that was a process called chemical evolution, which is not exactly the subject of our research, but of one of our neighboring labs actually in our building. And chemical evolution means that in this primordial soup, back in the day when the earth was vast and there was no life yet, there were still molecules, organic molecules as well, that in some process that's not entirely understood yet, these molecules came together to make the first molecules, the first chain molecules that could be the hallmark of life. So these molecules were actually capable of reproducing themselves. So that's actually the beginning of life, basically. Inanimate matter coming together in a way so that it has a higher order function, and this higher order function can be replicated and reproduced to make more of these very, very, very primitive life forms. And so that's also, again, still an open question. If that process led to DNA building blocks and then ultimately DNA, or to RNA building blocks and ultimately RNA, or other building blocks and ultimately other nucleic acids, then some way down the line, one of these processes must have favored the emergence of the chemical system DNA to store information in all organisms.
Yeah, I'm like these nucleic acids have some like certain properties that make them really useful for like the task they have kind of grown into, I guess, in evolutionary terms, because they can reproduce, or can be copied in the context of DNA, and they are pretty stable chemically, and they are carbon based, so they allow a lot of room for variation. But in a different sense, they are also quite arbitrary. Is there maybe a way for non-chemists to understand in which sense, like how specific these chemical structures are that are used in DNA, or if you could imagine there being like 10,000 other molecules that could pretty much do the same job, which would also be pretty interesting to know in terms of, does it have to be carbon based, or does it have to be like even this specific temperature range, and these kind of questions that come with it.
Yeah, so that's again a question where the link between the first inanimate soup of matter and sustaining life comes in, and that link is somewhere where we come in as well. So as I said, let's say DNA is a given and it exists everywhere. So that's today's point of view. Then again, as I said, what we try to do is question that necessity and build other foreign molecules. But of course, if you don't take DNA as a given, then there are other possibilities. So the work that we do can also be informed by very early chemical evolution. And so one of these scenarios that has probably existed is the RNA world. And that is linked to an entire field, the Rna World Hypothesis, that basically states before information was stored in DNA, it was actually stored in RNA. And again, you asked me to explain this in a non-chemical way. The difference between these two molecules is minuscule, but it is there. And because every repeating building block of these chain molecules looks ever so slightly different, and let's say red Lego bricks versus orange Lego bricks, the entire structure has then a very different surface, very different 3D structure, very different chemical behavior. So they are actually on a biological level then very different, even if you just introduce an ever so slightly chemical modification. And in this Rna World Hypothesis, there was a world before DNA was the universal storage system where RNA did the job, and because proteins were not around yet to execute the coded function, RNA also had to do this job. So it was really a workhorse, both coding for some function and the code itself then executing that function, then actually having some kind of, if you want to say biological function, although that's early biology, but some kind of catalytic activity, if that rings a bell to some listeners, some activity that it also had to do at the same time. And we see some of those molecular fossils still in our organisms nowadays, and there's a lot of hints at conserved weirdness in us that makes this very plausible. And yet other theories say that it was never DNA or RNA. It was something called TNA, which isn't based on a ribose sugar but on a threose sugar, so ever so slightly different. But because every building block is ever so slightly different, then the resulting chain molecule has a vastly different structure and function. But that's a very intricate network of primordial events happening, and it's also a very intricate network of today's research happening along these lines. And it's very hard to find truth in this field because it's you always move closer to something like the truth, and you could digress vastly in that process, but you will never reach it really.
So to get into the specifics of what kind of truth you are trying to find and finding every day in your research. So at what we've heard a lot about DNA and the different layers of looking at it. So but at what stage are you specifically right now? At what specific things are you looking at, and with what kind of experiments, with what kind of tools are you attacking the problem?
So me personally, I am working on one of these unnatural nucleic acids, but for another purpose. So I have before explained how the general field came upon in the first place and what our lab and other labs in the field try to answer. But if you want me to, I can also try to hint at what I'm personally doing, if that's what you're alluding to.
Yeah, that's what I'm alluding to. Like you or your group, and like to get a more realistic picture of like how can you actually tackle this problem of trying to understand these systems and build these systems.
Okay. So the very fundamental step in the process of building these systems is exactly that, synthesizing them. So it's a very physical process. Whereas in other labs that work by analysis and deduction, you gain insights, and that insight can be stored in your mind and on paper, and it will not change. But in our lab, you actually have to have something physically in your tube, physically in your reaction setup on your desk that can do something. That's quite exciting, actually. But it's also the limiting step. So in order to build these unnatural nucleic acids, these unnatural systems, you have to basically start from scratch. You have to take a red Lego brick and take blue paint and paint this Lego brick blue physically. So that's the very fundamental chemical synthesis step involved in all of that. And I have already done that for my system. So I've taken red Lego bricks and I have painted them blue, basically, so that I obtain the unnatural building block that makes up this unnatural system, this unnatural nucleic acid. But only having these Lego stones, these blue Lego bricks, doesn't solve the problem yet. So step number 2 is to be able to stack them on top of each other. And that's where these molecular machines come in that I have mentioned a couple of times, but I haven't said the technical term. I mean, I'm digressing so far in my answers and becoming very technical that I shy away from being too technical in my terminology as well. So I hope that my digressions still remain understandable by having understandable language. But of course, some terminology helps sometimes. And these molecular machines that are able to build these towers of Lego bricks are enzymes. And enzymes are basically proteins with a function, with a catalytic activity. And these enzymes that in all organisms perform this task are called polymerases. Polymerases are machines that are able to polymerize, i.e., make chain molecules of individual building blocks. And there are many other chain molecules, but for nucleic acids, for Lego towers, these enzymes are called polymerases. And the problem is that we have polymerases in our body that are able to chain together our DNA, that are able to stack red Lego bricks. But if you hand these enzymes blue Lego bricks, they fail. They just cannot cope with it. It's like it's like a crane, and there's a crane operator that has been told to collect red Lego bricks and make towers of them. And then you suddenly hand him a blue Lego brick, and of course he would say, "What? No, I've not been trained for that. I don't know what that is. I don't need that." So fundamentally, what you have to do is you have to retrain the crane operator. You have to also change other things about the crane's arm and the shovel and all constituent parts, so that this new crane that you then build, this modified crane with its slightly persuaded crane operator, will then happily stack blue Lego bricks. So that's step number 2. And you can already tell that without me actually saying what this means on a day-to-day basis. It's incredibly technical, it's incredibly preparative. So you have to really physically put different constituent parts together in a tube and hope they work. And the hoping part is of course not hope. We try to be as scientific as possible and not rely on hope, but rely on other very well-established techniques in the lab. And these techniques, if you want me to, again, I have to stop myself then here. If you want me to, I can quickly try to explain what this technique is, but it could lead too far.
I think what's usually, if you talk about these things in abstract terms, sometimes it's kind of easy to miss out how incredibly insane it is that we are as human beings actually capable of manipulating these systems so well that it works. Like you mentioned polymerase. Maybe that's also like a good example that connects to today's public health crisis with COVID.
Yes.
So the COVID tests are based on like our Pcr tests, which allow to take this viral DNA and copy it really quickly. So I don't know if you quickly want to mention PCR, because it's also an extremely important technology in the field. But then we can move back to what more specifically, what kind of experiments you are doing.
Yeah, absolutely. I mean, we can spend a lot of time talking about the fundamental science behind the virus SARS-CoV-2 that's currently rampaging in the world. But yeah, the process that's used to detect the virus is exactly PCR, which is short for polymerase chain reaction. So that already, it's actually nice that in the public conversation you use an abbreviation that contains exactly what I'm working with, that contains the letter P, the polymerases. So in PCR, you take one of these chain molecules, these nucleic acids, and you amplify it. So you copy it so many times that some downstream process is able to detect them. Because if you are infected, not you, but if one is infected by SARS-CoV-2, then the infected person's body will have the genetic information of this virus in their body. So what the PCR test is doing, it's trying to take that virus's nucleic acid, that virus's genetic storage molecule, and make so many copies of it with help of these polymerases that basically make copies and copies of copies and so many copies of that molecule, so that there is a high enough number that you can detect it. And if you had the virus molecule in you, the virus genetic storage molecule, then this test would come out positive. But of course, if it wasn't there in the first place, then the polymerases cannot amplify anything and it would come back negative. That's the fundamental principle of it.
So yeah, to get back to the previous question, so how do you convince the guy in the crane to play with your blue Lego bricks?
Exactly. So the crane, again, being the polymerase that's able to make copies and copies, just as in PCR. But the problem is in PCR and in our bodies and everywhere, these polymerases are trained to work with DNA and RNA, are trained to work with red and orange Lego bricks. So because we don't really know what we need to do in order to make them cope with blue Lego bricks, we essentially play evolution. So the technique is called directed evolution or in vitro evolution, which means evolution in a test tube. And you do exactly what evolution meant by survival of the fittest. So we basically produce a whole host of variants of these cranes, a whole host of variants of polymerases that are ever so slightly different than the natural counterpart, the templates that we come from. And we try to introduce all of these changes at a very untargeted fashion. I mean, there is maybe the good analogy of using a shotgun approach. So you use a shotgun, a molecular shotgun, to introduce at random all of these changes in the crane, in the polymerase. And then you, in one test tube, you let all of these new cranes, all of these ever so slightly changed polymerases, do the job of stacking blue Lego bricks. So you suddenly give them a new task. And the one polymerase, or the polymerases, the cranes that are more capable at performing this new task, this task of stacking blue Lego bricks, they will be physically fished out of the test tube. And so it is basically survival of the fittest molecule. The one molecule, or the molecules, the polymerases that are able to do the job the best, that are the fittest for what you want them to do in the future, they will be selected for, they will be fished out. So basically exactly what evolution did, but not over billions of years, but days and a couple of weeks. So you go through iterative cycles of having new and ever new polymerases stacking blue Lego bricks, and they get better and better and better. You basically train them to do it. You introduce changes that then suddenly make them better, and you continuously fish out, physically, like quite literally, physically fish out the fittest polymerases, and that's called directed evolution. That's what we use in the lab on a day-to-day basis.
That's pretty crazy, actually.
I think so too. I mean, it's exactly what got me started in the first place and got me interested in that. It's of course nucleic acids as a very intriguing and exciting system to work with, but this technique, directed evolution, is still blowing my mind to this day. And it was awarded, so the entire technique was awarded the Nobel-prize for chemistry. And to not say anything wrong, I have to know if it was 2 or 3 years ago. Time is flying, man. It was in 2018 when 3 scientists were awarded the chemistry Nobel Prize for this technique, for directed evolution. And one of them happens to be my PhD supervisor's PhD supervisor.
Nice. So yeah, standing on the shoulders of giants.
I didn't want to say it and come across as that. It's just it makes you. No, on the contrary, I think it makes me a bit humble because I have just years after this was invented inherited this technique, and we are using it on a day-to-day basis, and it's just mind-boggling what it can do. It's very powerful.
Yeah, I meant it more in that sense of like having these crazy, amazing tools like that were invented by just brilliant people. I think that's one of the things that makes science to such a joy, because you can just you're working
absolutely.
every day with some of the greatest inventions that people ever came up with. So that's kind of a luxury, I guess.
Yeah, absolutely. I mean, science is an incredibly collaborative endeavor. I don't know of many fields that are, or many endeavors, human endeavors that are so collaborative. Of course, everyone listening to this and being further down the academic line will say, "Oh my goodness, he's so idealistic," and wait until he sets foot in academia more. But of course, I cannot help at this point but look at it and that way and see how many insights actually have to come together and how many hours, days, weeks, years of research of millions of people and so many generations of PhD students and postdocs have to be accumulated and recorded properly and reported properly and understood properly by the next generation in order to give rise to today's research. That's really incredible to think about.
Yeah, and I mean, it's the influence it's having on us and our societies is pretty outstanding. So it's like it's also an enterprise that's really changing everything pretty dramatically. I mean,
That's right. I mean, because the steps that science takes are very small steps and they take a long time to take, it might come across to the public that science is this ivory tower endeavor by academics with confused gazes and crazy messed up hair, and it's something that no one else can dive into because it takes so long to get into, and then you don't change anything because everything takes so long. That's what it might come across it, but actually 1 second later, when you take a closer look and when you actually think about the things that we take for granted today, like a PCR test for a virus that's causing the world to turn upside down, when you just think about that for one more second, then you realize that there had to be a crazy guy called Gary Mullis at some point to invent PCR, which only makes it possible to detect this virus. So science is everywhere, and the insights of science, even though they take a long time, they really shape the world in every way. Sometimes to the negative, of course, and it's a very important discipline of a scientist to be ethical and to think about the implications of their research. But on the grand scheme of things, it just makes things possible that wouldn't be otherwise possible.
I mean, you're also part of a field with, like, roughly speaking, in the direction of genetics that is it's both making people very excited and also people a bit scared about what the future will bring. So
Yeah.
I think it's pretty clear to expect that genetics in the next 50 to 100 years will really transform how we approach a lot of things and maybe even how we view ourselves as human beings.
Absolutely.
So, and I guess we are also seeing right now with the COVID crisis that genetics kind of there's this big demand for like an improved understanding and new techniques from your field, starting with vaccines like that are also based on RNA. For example, these RNA vaccines also are kind of go in the direction of what you have been talking about, and they will also transform like the medicine of the future. In what sense can maybe in a very general way, what can you say about this, like how genetics affect society, what kind of stereotypes there might be about it, and what kind of you think the future will bring?
Genetics as a word tries to cover a field that cannot be covered by one word. I mean, I am not a geneticist, right? I'm a chemist who happens to be working with genetic molecules, but I'm in no way building any cell that is living on its own and can do things that it would not be able to do without my work, right? I'm not constructing a new living being. And in the same way, genetics as a whole, or geneticists, are, of course, always in the spotlight of public conversation. I mean, when you hear the term genetics or genes, then what immediately comes to mind is this unfathomable world of information that some weird scientists in their chambers are able to meddle with, that has crucial consequences, but that nobody really understands. And of course, in some ways that is true. We understand very little about the interplay of all of our genes, especially in humans, because there's so many genes that have such an intricate network of function. But that doesn't mean that there shouldn't be more and more research into this. And what the public might think about genes is that they are bad inherently. They are non-GMO products advertised as such, genetically modified organisms, GMOs, and products that don't contain them, that would never contain them in the first place. I mean, there is food in the supermarket that says it doesn't contain any genetically modified organisms. Well, that's because it doesn't contain any organism in the first place, no matter if it's modified or not. Or the other way around. There are products that are advertised as not containing any genes, whereas genes are everywhere. A tomato has genes, and quite many at that. So it's incredibly insane. So, and the fact that our tomatoes are ripe and red and stiff and appreciative looking and pleasantly looking is also due to the fact that generations of humans have crossed different strains of tomatoes to select for new tomato strains that have better properties. And that's genetic engineering, that right there. It's just not engineering in a very targeted, concentrated manner, but it's basically changing the genetic makeup of this plant. And so everything contains genes, and it's nothing to be scared of. On the contrary, it's everything. I mean, life is genes. Like everything that is living is based on genes and their interplay. And this doesn't have to be frightening. No, on the contrary, actually, it can be empowering, because as soon as we understand more and more about genes and how they are, what they're looking like, what our human sequences of these genes are, and what they cause, the more we can actually try to cure diseases that we couldn't cure with non-genetic means, for the lack of a better word. So there are medicines of the future that are very reliant on our understanding of genes. And maybe listeners of this podcast have heard of CRISPR-Cas, which is already in widespread use across academic labs, and everyone is using it. And it has, of course, dangerous implications. And at the same time, I don't want to say but, and at the same time, huge potential for changing diseases that are, or curing diseases that are due to faulty genetic code and genetic mutations. And if you want me to, I can talk more about it, but I'm going to bring this answer to a close here.
Yeah, sure. We can maybe like the medicine of the future aspect and how genetics might transform medicine, I think is extremely interesting, and also something that is becoming apparent again at the COVID crisis that we already mentioned a couple of times. That is so constantly on everybody's mind. That, for example, if we analyze the genome of people that are especially receptive to have a strong COVID symptoms and to have potentially life-threatening COVID systems, then from that genetic analysis we can figure out what happens, like what COVID is actually doing in the body. And from that also come to potential treatments. We already before talked a bit about COVID treatments, and you mentioned remdesivir and how it works in the cell. So can you elaborate a bit how it functions and how that relates to maybe how that relates to how a virus in general works and how it attacks the cell and how it reproduces?
Oh, okay. Yeah, I can do that for sure. Then I'd have to answer the latter question first, which would be how does a virus work in the first place?
Yeah.
Well, Manu, you're triggered. I mean, I cannot not digress with these questions.
I mean that's the point of the podcast. It's endless digression for the fun.
Perfect. Oh, who doesn't love a good academic digression? Viruses are as an entire class incredibly interesting. So first of all, viruses are not living beings, and I don't say this is ultimate truth because it isn't, and there's still a debate on this, which is interesting because viruses have been our companions forever, and still to this day, the vast majority of scientists would say viruses are not living beings. But the fact that this debate is not closed yet shows you how complicated it actually is. So there are many different forms of viruses, but they are all composed of some form of shell, some form of capsule, some form of compartments in which the virus carries its genetic information. So that's why it's actually complicated, because viruses are not living, but still they have genetic information, and that looks different for every virus. But in getting back to the virus that's causing the disease COVID-19, which is the virus SARS-CoV-2, this virus has obviously a very unique genetic setup, a very unique set of genes, which makes it have the phenotype that we see in today's crisis. So it is very infectious, and it's still not as deadly as other viruses, which makes their hosts survive very often, which is very beneficial for a virus, because if their hosts survive and propagate virus in them, then viruses can jump to another host and from person to person. So viruses are not living. They are a form of parasites. They cannot live by themselves. They cannot replicate. That's one of the hallmarks of life, replication. Viruses cannot replicate themselves alone in thin air, in water, wherever. They need need to form a parasitic relationship with a host that they infect. So, and viruses are very specific. Many viruses are very specific for the host that they need. So, SARS-CoV-2 came many organisms down the line from bats. And now that SARS-CoV-2 spreads from human to human, it has changed vastly. It has changed its genetic makeup. It has evolved its genetic makeup to be very infectious among humans. And so that's the set of genes, the information that's unique to SARS-CoV-2. And then it needs to do something with these genes, right? They are just lying there in this capsule, but SARS-CoV-2 alone, as one particle, is not able to do anything with it. It's not able to live and replicate on its own. So it needs its host, which means it is docking onto a human cell. So if people have seen depictions of viruses, then, and especially coronaviruses, then they realize that there are these spikes sticking out of the surface of the virus. And these spikes, these crowns, these coronas, are very important to functioning of the virus. So these spike proteins, these needles sticking out of the virus, they dock to a receptor called ACE2 on human cells. And after this docking process, the virus is internalized by the human cell. It opens up and it releases its genetic information. It releases its nucleic acid containing all of the SARS-CoV-2 genes on it. And in this case, it is an RNA molecule. SARS-CoV-2 are RNA viruses. And one of the genes that it encodes for on its RNA is a polymerase, which upon production by the host cell is able to make many copies of the viral genome. So that's exactly where it's important to have the host and to form this parasitic relationship, because the virus needs the machineries of the human cell to produce more of its genetic material and to produce in the long run. So it needs the machinery of the human cell to read its own genetic code, basically, and to produce this polymerase. And the human cell cannot differentiate between the RNA molecule that comes out of a SARS-CoV-2 viral particle and the RNA that it itself has produced during transcription, which, if you remember, was the first step of the central dogma. So the first step of transcribing DNA to RNA. So this messenger, this human middleman, this RNA molecule, looks exactly the same as the RNA molecule that is blasted out of the virus. And that's due to our universal chemical language of these molecules, of these. That's exactly why we can be infected by viruses, because their genetic system uses the same language as our genetic system. And that step that I just described, where the viral RNA is read by human machines to produce the virus's polymerase, that step is exactly where drugs come in to treat the virus, to treat humans that are infected by this virus. So the crucial agent, the crucial machinery that the virus produces, that is produced for the virus in the very first place, is this polymerase that is able to produce copies of the viral genome in the human host cell. And the drugs that are used to treat viral infections, they are basically Lego bricks that are disguised as natural red or orange Lego bricks, but they are not quite. So if you have a Lego brick, you know that it has these pins on the top and these holes on the bottom, right? Obviously, because you need to stack them to a tower. And a viral treatment like remdesivir would be a red or orange Lego brick that has pins on the top but no holes on the bottom. So the viral polymerase that us humans have just produced for the virus ever so generously will then accept this disguised Lego brick and put it into the new chain that it's currently producing. But at that very point, it stops because there are no holes on the bottom to put new Lego bricks onto. So basically, you make the viral polymerase fail by giving it a disguised, faulty Lego brick that's not functional anymore.
Yeah, that's also pretty incredible. Get to see how important it is for us to understand like how these things actually work. And I recently I read this article about kind of how modern science reacts to the COVID crisis versus how people in the 14th century reacted to the plague. And then like back in the day, that like the biggest scientific institution of the world, like like the university in Paris, which was obviously pretty strongly influenced.
Yes,
they deduced that Mercury was in and Saturn were in a bad constellation to each other, so that's why the plague was coming down on Earth. So I think it's pretty reassuring to see that like the vocabulary in which we can talk about what is happening with COVID has like kind of improved a bit over the years, of the past years.
But exactly, it's not a plague that's posed onto us by God or by the constellation of the stars, but it's actually very well or slightly well understood process of a virus wanting to live, wanting to find new hosts that are very sociable and hosts that like to come together and give virus from one to the next. But understanding that is obviously crucial, and the vocabulary might change again, and generations of scientists further down the road, they might look at us the same way that we look at the early academic institutions or the church trying to rationalize what is happening. But that's just inherent to the process, I guess.
Yeah, but that's also an interesting question, like about your field. All science is always like moving along on the edge between the known and the unknown. So, and with the COVID crisis, obviously, we have also seen that there's a lot of things we don't understand, and we still don't have like a fully causal model what COVID is actually doing in the body, and we still don't have any functioning, really, medications and no functioning vaccinations. So there's still a lot of unknowns, even though we have better causal models. So how would you say in your field and with what you're doing specifically, it's like really like moving around in the dark, or is there a certain paradigm and framework that you work in that's really well established, and you're more like working on really specific things? Or maybe it's also a mix between the two, I guess.
Yeah, it will always be a mix between the two. So the fact that in my field now, the fact that DNA looks the way it does, chemically speaking, and does what we see it does, that is well established. But we constantly learn new functions and learn new things about our genetic molecules that we didn't know before. So, for example, the middleman molecule, the messenger RNA, it has so many more functions in our bodies than just being the middleman, and they are still researched. There's still more insight that has to be gained on that. But nobody would debate the truthfulness of the X-ray measurements that Rosalind Franklin had done back in the day that led to the model of our DNA double helix the way it looks now. So we are building on solid ground, although in our science there is never truth. There's only truth in mathematics, but still we are building on solid ground and a lot of evidence that it is actually that way. But then from there on out, as I tried to explain earlier, synthetic biology is in essence digging in the dark. That's what it makes. Yeah. That's what makes it so interesting, that you try to use techniques like directed evolution and you try to use synthesis to build something that hasn't been there before. You try to use existing material or unnatural material to build unnatural function and equip molecules with new function. And so that's the nice thing. Every step forwards in our field is a synthesized molecule that hasn't been there before. And if it's interesting is another question. If it's good at what we want it to do, that's another question. But every molecule we produce, it doesn't matter if it is made of foreign or natural building blocks. Every molecule we produce potentially has a new function, but that in essence is digging in the dark. Directed evolution in essence is not knowing where to end up, but trying to introduce all of the paths that you could possibly take to possibly end up with one molecule that can do something new.
Yeah, that's pretty much how nature does it. It's all
exactly
kind of trying to, in the dark, randomly hope for the best, and then
exactly
suddenly human beings come out.
Yeah, exactly. Only nature would always change one thing at a time, and then if it's successful, the organism survives. If it's unsuccessful, the organism dies and does not contribute to the future of evolution. So that's how nature does it, in a very untargeted fashion. Whereas we also do it in an untargeted fashion, but we try to speed up the process by introducing all of these changes at the same time.
Yeah, you don't need all the dying and pain and suffering to get progress with your
although suffering we do need.
Yeah, indeed. Yeah, that's there's no other way. So mentioning all the suffering, like how does everyday life in your research look like? How does a scientist spend his day? Like as specific as you want to get.
Okay. The unspecific answer would be coffee break, lunch break, coffee break, home. And while that is partly joke and partly very true, there's also some more fundamental truth to that. So a lot of science is actually talking about it and sitting together with your immediate colleague in the lab or with the entire lab. Everyone has their own project, but we all work with similar techniques and have and encounter similar problems. So talking about these problems oftentimes leads to very insightful solutions, in insightful just discussions. And then once you're done with all of your coffee and lunch breaks, you have to go to the lab and actually do it. So there's no way around actually doing it, which I like. So our science, our lab-based science, or as we say, wet lab-based science, is very hands-on. You literally get your hands dirty, and or your gloves dirty, of course, if you're properly equipped with personal protective equipment. But there's no excuse to not do things, and you just have to go ahead and put the interesting ideas that are based on caffeinated discussions into practice. And so that's very interesting and makes it very exciting. In a way, I get up in the morning at very irregular times where every person working in industry would shake their heads and go to the lab. And after the coffee breaks, the inevitable coffee breaks, I try to test one new idea at a time, maybe sometimes even 2 ideas in general. If I'm very crazy, I would do 3 things in parallel, but you can just, you see how your mind dilutes and you cannot really focus on one thing. Because once you have done something, you get some data, you get some result. For example, you try to equip a crane with new function, and you have just blinded the crane operator. And then you have to know if that leads to the fact that it doesn't pick up any Lego bricks anymore, or if the blindness actually leads to the fact that it can now incorporate all Lego bricks. So once you have that result, you need to analyze it. So a lot of everyday lab work is also sitting down at a desk, staring into a computer and analyzing it. And the means of analyzing your data vary. Some people in our group actually code, they program their analytical workflow, and other people look at spectra that tell them if their synthesized Lego brick looks the way it does. That's what I do, for example. Or they look at the activity of the crane, and how well it operated now that you have made the crane operator blind. So it is a cool interplay between talking about science, doing science, and analyzing your science, and then planning the next step and planning what you want to do the next day, the next week, the next month.
So what would you say are like maybe the best thing about working as a researcher and the most unbest thing? Like what kind of what makes you get up in the morning with joyfully, and what makes you want to stay in bed?
By the nature of me, I would start with the bad news so that I end up on the positive note. And I mean, if there's anyone in academia further down their PhD or even in their postdoc, or even, God forbid, a PI listening to this, a group leader, they would probably think, oh yes, what a lovely first year PhD student, still so enthusiastic and so idealistic. But one thing at a time, right? I mean, you ask me now, and you can ask me again in 2.5 years, and then we'll see. But let's start with the downsides. As I said, things take time. If people are impatient or they need the instant gratification, and of course all of us do to some extent, I don't want to speak myself free of that, but if you need the instant gratification of something you did, some instant result that's ideally positive, then science is not really the place to be. And of course we're all on the spectrum on that, and I would of course get frustrated at failing experiments, or experiments that take long detours and long alternative routes that I have to take in order to make it work. Because, as I said, it's very preparative, and in order to get to a physical molecule in your test tube that you can then show to the world, you have to go down a lot of steps, and that is never a linear way, never ever. And before my PhD, I was writing these project proposals for my applications, and they look so linear and they look so logical, and it never is that way. Never. I mean, I've already digressed vastly from that, and luckily it's going well. But of course, the actual routes that you take is always different. So things take time, things take detours, and things fail a lot. So you have to know that this is that way, and you have to make peace with it. Maybe even kind of indulge in the fact that it is a constant learning. It is constant failure and then being informed by this failure and then retrying in a better way. Or some scientists, I should know who said that, but sometimes scientists said, well, you just fail, so next time fail better. That's exactly what it is, basically. And so that brings me then to the positive things. It is constant learning, honestly. That's the nicest thing about science. As a researcher who is still in the lab, or as a group leader or professor who has their own lab and is teaching a new generation of scientists, it is constant learning. It never gets old. You never get old. You're always surrounded by young, ideally enthusiastic people who want to produce something that hasn't been there before, because that's academic work in essence. You produce something that hasn't been there before, that cannot be found in the textbooks. So whereas before entering science and your own research, you learn what people have done before you, you then use this knowledge to produce something that hasn't been there before. And as idealistic again as it sounds, it is exactly what makes me go on and go on and go on, because even though it's just an ever so slightly small dent in the whole wealth of knowledge, it still is a dent. It still is some furthering of what we know. And even if someone's research fails completely, that already is a furthering. You still know what doesn't work. And of course, you would want your project to not fail, but be successful. But and then you would produce some kind of furthering, some kind of dent in the human knowledge and progress it in some way or form. And so, yeah, it is constant learning, but in a very applicable fashion, where you constantly know more about your system. You constantly learn about it, and at some point, you know more than anyone else. So, like, it is your personal baby, and you get up for your baby in the morning, and you nourish it and nurture it, and it has to progress and evolve and grow, and only you know best what to do with it. So that is quite exciting. And then surrounding yourself with other people that do the same thing, which of course is not so possible in these days, but surrounding yourself with other researchers that maybe work on a similar thing or on a related field or something completely different, you then feel this communal spirit of wanting to put all of these babies together and actually produce many dents in human knowledge and further human knowledge, not one step at a time, but maybe many researchers at a time, at the same time. And to be a part of this progress in time, to be part of this culture, is very exhilarating and very fulfilling as well.
That's a beautiful answer. So is there one like big lesson you learned, would you say, in the last 1 or 2 years of like really professionally getting into research? It's like one big lesson you would give to people that are in a position of thinking about going into research or thinking about doing their PhD, and like thinking for themselves, like something that would have been nice to know, I guess.
Well, everyone is tackling this a different way, and everyone's persona is different. And I would say, being the way I am and having made the experiences that I have, I would say science is a very emotional endeavor, and at first it might not look like that, because scientists need to be analytical, and science is a very analytical, rational endeavor. But getting into science and staying in science and persevering in science is a very emotional endeavor. and that also holds true to getting into a PhD. I would say to people that want to do a PhD, people that want to get in science, whatever phase in life that would be, it doesn't matter. I would say you have to be emotional about it. You have to know that you want to do it. You don't have to know that you want to do it. You have to feel that you need to do it. Actually, that's the essence, because science is hard and it takes time and it will fail, and you will not persevere through that if you don't feel it. You don't have to be super intelligent or ambitious or accomplished. You have to persevere above all, I think. And perseverance is fun if you know what you're doing it for. And it's not fun. It's meaningful. It's your scientific endeavor is meaningful if you do it out of an emotional necessity. And then also finding your field of research, finding your lab that you want to work in, that should, in my opinion, also be a very emotional endeavor. And you should think yourself into the future and try to see what it feels like to work in this lab. Try to think about what it feels like to work with these people, and if you can survive, emotionally survive all of the hardship in this field, in this lab, and if you are willing to sacrifice other things and go all in and feel it and be emotional about your research and do it because you need to do it and not because you think you have to do it, then that's the way to get into science, in my opinion.
Yeah, that was a beautiful answer as well. So I think there's probably no better note to end this on than this one. So I'm extremely grateful for you to take the time and have this fascinating conversation.
It was
with
an
me.
absolute joy, really. Thanks for having me. And when we first had the idea, I already thought it was a great idea. But then I always get swept up in the process and don't realize how 1.5 hours pass, and that tells me that it's just it's great to talk about it.
Definitely. So thanks again, and
thank
have
you.
a wonderful
You
day.
too.